Hello, everyone. My name is Kalle Simpson. I am a senior at North Carolina State University, majoring in environmental science with a focus in marine ecology. And today I will be presenting research that was conducted this past summer as a part of an NSF REU at the Moat Marine Lab in Sarasota, Florida, where I interned under the mentorship of Dr. Eileen Maldonado in the ecotoxicology lab. So my research entitled Oxybenzone-based sunscreen alters cortisol levels and behavioral patterns in sheepshead minnow focuses on the impact of the tourism industry and the fisheries industry. So I'm from a small town in North Carolina that relies on both of these industries, and I could see the parallels in Florida as well. And I wanted to understand how these industries are interconnected with the environment as well. So oxybenzone is an active ingredient in many sunscreens, and we know that it's harmful to coral reefs, but not many studies have been done with oxybenzones in fish, more specifically with sheepshead minnows. So oxybenzones are endocrine disruptors. They can cause decrease in fertility and a loss of territorial behavior in fish. They're also lipophilic and bioaccumulate up the food chain to secondary consumers and game fish as well at the top of the food chain. So I also wanted to know if these oxybenzones altered stress levels in fish, because heightened stress levels can result in slower growth rate and an increased susceptibility to predation, which could again decrease population and alter the food web and could ultimately hurt game fish, which would hurt the Florida economy as well. So in order to test this, we measured out 4 treatments which used 4 different doses of oxybenzone. So we used 0 milligrams per liter, 0.005 milligrams per liter, which is the environmentally relevant concentration, which was found in a study by the International Coral Reef Initiative, 0.05 milligrams per liter, which was a nice medium, and 5 milligrams per liter, which is an extreme high to definitely elicit a response. So these were measured out every day and dosed, and then the fish were recorded for abnormal feeding and swimming behavior. Each tank was recorded for 5 minutes, and each tank had 2 fish, so that would make 5 to 6 fish per treatment. So abnormal feeding behavior was categorized as fish being aggressive, disinterested in food, or just not interested in finding any food in the tank. And abnormal swimming was fish exhibiting erratic swimming patterns or little to no movement. So after 72 hours, the fish were then they were euthanized, homogenized, processed through a ELISA cortisol kit. So moving on to our results, we can see that the highest percentage of abnormal swimming behavior was at 60%. And this was at the second highest dose. And in the video, you can see fish kind of like thrashing around and twisting their bodies, which I thought was pretty interesting. And then we can see this high dose exhibiting 50%. And after this day, mortality was at 100%. So that's why there's no more data. But we can see that at least 2 of these doses exhibited abnormal behavior as well. And moving on to feeding behavior, this day that we see the 80% fish, they just did not care about any food in the tank that day. It could be because their visibility was so low. Again, that's another study that could be done on top of this, but we can see that also every day there is at least 20% abnormal feeding behavior in many of these doses. For the survival rate after day 2, so after 24 hours, there were no surviving high dose fish. As the dosage increased, the survival rate decreased, so we have an inverse relationship there. And with our cortisol absorbance, we actually had to take a ratio of the protein to the amount of cortisol in their bodies because we had some issues with the ELISA kit. So rather than being a quantitative measurement, it's more of a baseline observation kind of ratio. So we can see that there was some variability in one of the doses, but these higher doses had the highest amount of cortisol absorbance in the fish. So to conclude, behavioral patterns were altered at every dose of oxybenzone, and the mortality rate increased with dosage. The lowest dose, the environmentally relevant one, exhibited a low mortality rate, but low doses had a higher rate of altered behavior over this time period. And finally, cortisol absorption followed the same trend of increasing with the dosage of oxybenzone.